This is going to pick up at the cell cycle, so Roman numeral 3 on your outline. The cell cycle is everything that involves the cell dividing. So let's take a look at the parts of the cell cycle. We have the cell mostly spending its time in something called interphase. So interphase is the part where the cell is doing its job. It is making proteins, it's doing whatever it needs to do to housekeep, to make sure that it's healthy and maintaining homeostasis and all that good stuff. So 90% of the time, the cell is in interphase. Then, when the cell wants to divide, it's going to undergo mitosis, or enter the mitotic phase. This doesn't take a lot of time, 10% of its time, but it is a very dynamic time with lots of subphases. So let's take a look at the pieces of interphase. So you can see G1 here, that stands for gap 1. In gap 1, the cell is dividing, the cell is getting ready to divide if it needs to. It can bulk up on nutrients and preparing metabolic processes to divide. In the S phase, the second phase, this is the DNA synthesis phase. So S stands for synthesis. So when the cell is going to divide, it's going to divide by copying its DNA first and then dividing the cell second. So we have chromosome duplication. Now when you see this image right here, this is one chromosome. This is one molecule of DNA that has been wrapped up into a bundle, right? Remember the word supercoiled. It's been supercoiled into a chromosome. So when you copy a molecule of DNA, it's going to look like this, right? The two identical copies of the chromosomes are going to be next to each other. And this is going to be apparent very soon, but this is always what happens. The exact copies are going to be attached to each other. They're attached to each other at this central point here called the centromere. And at this point, each one of these DNA chromosomes are called sisters or sister chromatids. Okay, and then there's another gap phase, the gap 2, where the cell is still just prepping to divide. It is undergoing normal processes, but it's also preparing to divide. And then there's the actual physical division, where the two identical copies of DNA that we just made in S phase are pulled apart. Two cells are formed, and one copy goes into each cell, and then we we can go do the loop over again, or we can come out of the reproduction loop into a resting phase. Okay, so this is just to further talk about the DNA replication. So this is one DNA molecule, one chromosome. This is still that same chromosome, but it's been duplicated, and the copies are attached to each other at this middle point that comes in. It doesn't have to be in the middle, but it's a point that comes in. This is called the centromere. And then during mitosis, when the two cells are being created, each sister chromatid goes to one cell, and the other sister chromatid goes to the other cell. So that way the new cells that are created, the daughter cells, each have a complete copy of DNA. And this is just a cartoon. Dude, mitosis starts in 5 minutes. I can't believe you're not condensed yet, right? So in order for the cells to copy DNA and to put them in different cells, they have to be supercoiled. So this represents chromatin, and then chromatin condenses to form a chromosome, right? So this is a picture that we saw. All right. Cytokinesis is a process that follows mitosis, or it's associated with the last phase of mitosis, but it's literally cutting the cell in half. So you can see that we have one cell here, but it's starting to really form a deep groove down the center. This groove is called a cleavage furrow, and the cleavage furrow will deepen until the cell splits in two. So what you see here is what's happening right here, and the cell split in two. And then this is another image of the cell splitting in 4. So the 2 cells are then splitting again to make 4 cells. I will post these videos that I would normally show you in class in the module for this chapter, so you can watch them through the module. So let's talk about the phases of mitosis. So they are prophase, metaphase, anaphase, and telophase. So if you look at letter B in your outline, you need to be able to recognize what goes on in each phase and to recognize what each phase looks like in a diagram or in an actual picture of the cell. Okay, so that's what you're responsible for. So let's start with what's going on in interphase. So remember, interphase was what the cell was spending most of its time in. Let's look at what's going on in both of these images. So our DNA is in chromatin form, right? So there's the word chromatin. It's duplicated, so because S phase occurred. This is a picture of G2. Remember, S phase came before G2. So the DNA has been copied, and we are now getting ready to divide. There is an organelle here that I have not mentioned to you before. This is called a centrosome. A centrosome is two of these little sort of cylinders next to each other. So what we have here are two centrosomes. The other word that is used for this organelle is a centriole pair, because each one of these little cylinders is a centriole. So a pair of them is a centrosome. So it's a little bit confusing maybe, but I tend to use the word centrosome. So we have these organelles called centrosomes, and these are really going to be important because they're going to grow spindle fibers out of them. So you can see what's happening here. These spindles are going to organize and push and pull our chromosomes to each other. So it is up to the centrosome to set up the two sides of the cell when the cell divides. And it's going to allow those chromosomes to move. So things that you should notice in interphase. Number one, we have chromatin. Number two, the nuclear envelope is still intact. Number three, we have two centrosomes still sort of at one end or one pole of the cell. In prophase, what's going on? So let's take a look at the actual picture. So what I can see is that the nucleus is blue. I don't see chromosomes. I don't see anything. All I see is just like a nice sort of clean blue circle. I see the green. These are going to be the centrosomes that are starting to sort of ramp up the growth of some fibers. Everything in red is the rest of the cell. In prophase, we can see some rapid things happening. So I'm going to group these two in the same prophase. What's happening is the chromatin has become chromosomes. So that's number one. In prophase, chromatin, or letter A in our notes, chromatin becomes chromosome. So it's supercoils. And you can see this. This is the first stage, prophase, in which you see the chromosomes. We can see the spindle fibers in green, and we can actually see in yellow those two centrosomes. Number two, or letter B, the nuclear envelope begins to break down, right? Because if we want to access our chromosomes, then we need to break through the nuclear envelope. It actually breaks down. Letter C, the centrosomes double, begin to move the opposite poles. Spindle fibers will grow out of the centrosomes and physically attach to the sister chromatids at the centromere. So that is more apparent in this image. Look at the spindle fibers that are growing, these fibers, and look to see where this one's going, right? It goes smack dab to this centromere on this chromatid, this sister chromatid, which is facing this pole. Look at the other side. There's another spindle fiber that comes up and grabs onto this another sister chromatid, right, the opposite side, and it's going to attach to the centromere from the opposite side. So now you can see that the centrosomes, their job is to set up the two ends of the cells, and they're also growing these fibers out. So this side is going to attach to all the sister chromatids that are facing the bottom, and the top centrosome is going to grow and attach to all the chromosomes that are facing the opposite side. So we're going to set up this situation in which we can start to move this chromosome unit around. All right, so the thing to notice here is that we can see our chromosomes. In the next phase, metaphase, what happens is these spindle fibers are going to push very dynamically and pull until all the sister chromatids, until all the chromosomes here are lined up along an equatorial plate. So this is an imaginary line that runs down the middle of our cell. That's called the metaphase plate. So when all the chromosomes are aligned, this is called metaphase. The next phase is anaphase. Anaphase, actually before we move on, let's look. So the chromosomes are in blue. You can see how they are generally lined up in the middle. They are long still, so they're going to look like long eyelashes or something that kind of stick away from that midline, but they're generally in that midline. In this anaphase, the next phase, these spindle fibers will start to shorten. Okay, the sister chromatids will separate because the spindle fiber attached to this sister chromatid is shortening, and it's pulling the sister chromatid away from the other sister chromatid, which is attached to the other side spindle fiber, which is also shortening. So because they're shortening, they're ratcheting the sister chromatids apart. So anaphase, the hallmark of anaphase is that the sister chromatids are separating and they're moving to opposite poles. Here we can see the blue chromosomes moving to those, and there's a space between them, right? This is clearly how anaphase looks. The last phase of mitosis is telophase, accompanied by the same time by cytokinesis. So in telophase, what happens is the cell is starting to set up to go back to normal. So each side of that cell is going to have one complete set of DNA that is going to start to unravel to form chromatin again. The nuclear envelope is going to reform, and each cell will only have one centrosome, which is the normal number. Meanwhile, the cytoplasm of the cell is going to start to divide. So in an animal cell with no cell wall, the membrane will start to pinch in, right, and form that cleavage furrow, and then continue sort of pinching and pinching until the two cells completely separate. And then we have two identical cells, or two clones. All right, so let's look at some pictures of mitosis in an animal cell. So here we have interphase. I know this is interphase because I don't see distinct chromosomes in my nucleus. In prophase and prometaphase, remember I'm grouping these two together, the hallmark difference between interphase and prophase is that I can see distinct units. I can see my chromosomes, although there are no particular sort of order. In metaphase, the chromosomes are very clearly lined up along that metaphase plate, so that's easily identifiable as metaphase. Anaphase hopefully looks pretty identifiable as well. I can see that the sister chromatids are separating to opposite ends of the cell. And in telophase, we have two cells now, and we have the nuclei of those two cells forming again, and those visible chromosomes are gone. They are now going to unravel and become chromatin once again. So that is a great image for you guys to remember, and you should be able to identify images of cells or diagrams of cells and identify the phase. Here's a third concept check. And this is our plant cell mitosis image. So plant cells are different than animal cells in for this, the stark difference is going to be that cell wall, because animal cells can pinch themselves in half, but plant cells cannot. Plant cells are going to build a new cell wall between the two new nuclei. So the sort of very small beginnings of a cell wall is called a cell plate. The cell plate will then completely divide those two cells together to make a new wall. So that's the only difference. Plant cells have to build a plate and then create a wall, but animal cells do not. This is a great image of plant cell mitosis, the actual cell. So we can see interphase, prophase, I can see chromosomes for the first time, metaphase, anaphase, and telophase. And now look, telophase, I can see that little cell plate, right? Because it's a plant cell. All right, this is something that I would do with you in class to have you guys try to identify phases of mitosis. But if you wanted to try and guess them and then check your answers down here, I'm going to let you do that on your own. And then here's another concept check question for you to submit for extra credit. And then here's another one. And then I was talking about cell cycle regulation to end this. So now, normal cell division has to be very tightly regulated. There are many, many checkpoints. A checkpoint is a molecular checklist that the cells do to make sure that they're not proceeding with a damaged DNA, or they're not proceeding, maybe if all the spindle fibers were not attached to their sister chromatid. You know, that would be bad because then one cell would be missing a sister chromatid. So there's checkpoints to make sure everything's working correctly. So the checkpoints are major messages to the cell. If there's something wrong, stop cell division. If there's too much DNA damage, stop cell division, because we don't want a damaged cell to keep going and dividing. So this is the normal process. There's a word here, apoptosis. That is programmed cell death. So if a normal cell can detect damage, and if that damage is bad enough, it will undergo cell death. So there is a neat, discrete way to die. The cell knows that its harm is going to be harmful, so it's going to kill itself, basically. So that's the normal process. In cancer, cancer happens as a result of mutation buildup in the DNA. So if you're exposed to a lot of UV radiation, for example, that UV radiation harms DNA, causes mutations, and those mutations can occur in these checkpoints. And if they're at checkpoints, if you knock out a few of your checkpoints, then you're in danger of a cell dividing uncontrollably. And this is that image. One cell that's damaged divides, divides, divides. It doesn't know when to stop, and that is when you get a tumor. So when cells are transformed, the word transformation is that process when a normal cell becomes a cancer cell. And that's through many, many hits or many, many mutation events that happen and build up inside of a cell. When is a tumor considered benign versus malignant? So a tumor is a mass of cells. It's already an irregularity. There should not be a tumor inside your body. However, benign tumors are masses of cells that don't go anywhere. So they grow to a certain size and they stop for some reason and they just sit there. So there's no danger of it spreading to the body. That's a benign tumor. A malignant tumor is it's a mass of cells that divides, and then for some reason that people are still trying to understand, the cells can break away, travel through the bloodstream or travel through the lymph channels and enter into a completely different organ and form another tumor. So that's a malignant tumor, and that's spreading. That is what they consider cancer. So a cancerous tumor or cancerous growth is something that can spread, and that spreading is metastasis. Metastasis is the process of which a cell breaks off from a tumor, travels through the bloodstream, and sets up another tumor elsewhere. And you've probably heard of really sad stories where someone's cancer has spread throughout their entire body. So that the original site of cancer could maybe have been breast cancer or bone cancer, but since it metastasized, that breast tissue has invaded the liver or invaded the bone or invaded the brain, and it just sets up the entire body to be riddled with cancer, which is very, very unfortunate and sad. But that is the basis of cancer. So the cell cycle, what you just went through, the mitotic cell cycle, is tightly regulated for good reason, because we don't want cancer to arise. This is an image of that breast tissue I was talking about. So if there's a tumor that arises in the breast and grows, it can invade the normal tissue, causing problems in that normal tissue. But then metastasis can also arise, where the cells will enter into lymphatic vessels or blood and go to other parts of the body. All right, so leaving on a happy note, cells are very resilient. If you transplant a piece of liver tissue into a patient, that piece will grow until it reaches the correct size and then stop. And in your body right now, there's about 2.5 trillion red blood cells in your body, and to maintain that number, 2.5 million new ones need to be produced every second by your bone marrow, and this occurs in a regulated manner, never overproducing or underproducing red blood cells. So your body is pretty amazing. These cells are pretty amazing, and they can self-regulate for most people's lifetime.